I'm dealing with much more disrespected organs like the mouth, in particular the salivary gland, which is probably the Rodney Dangerfield. We don't see it, doesn't do much since we don't think a lot about it. You know, it is the gland in your body that produces saliva or spit, and obviously that's important in moistening your mouth, aids in speech, as you all know. If you're giving a seminar or in a classroom and can't talk, that's because you have dry mouth. Obviously it's important in digestion. You got to swallow your food. It has to get to your stomach, the alimentary canal. It actually provides a lot of enzymes. They're important in digesting your food. And also, you have a lot of antimicrobial peptides that are produced in saliva that help protect your mouth. So why are we concerned about the mouth and the salivary gland? Well, there are also a lot of hypofunction or dysfunction of the gland that occurs. As you age, you produce less and less saliva. If you're on beta blockers or certain, actually antidepressant drugs, you develop dry mouth, xerostomia. And as both we grow older and as more and more patients are treated with drugs that affect saliva production, it becomes a bigger problem. Where it really is a significant problem is in patients that are being treated for head and neck cancer. They usually, you remove the tumor, but you also give whopping doses of irradiation to kill the tumor. But in the process of doing that, you tend to also destroy the salivary glands. Okay, so typical head and neck irradiation protocol is 5 times a week for 6 weeks, maybe more. And although people have developed very specific ways to target beams to try and kill the tumor and deliver high doses of radiation to the tumor, you still often end up with xerostomia due to this irradiation, inadvertent destruction of salivary glands. So you lose someplace between 50 to 75% to the production of your spit. Obviously, you got a real problem because it's lifelong loss. So people have tried to prevent this by giving drugs that cause you to release all your secretory granules. People have actually, after a patient has xerostomia or an experimental animal, they've tried doing gene therapy to put back molecules that stimulate the secretion of water, aquaporins, and those really aren't very effective at all. And we got interested in this in a very inadvertent way, and that was that we were involved in a program project grant with people in the dental school looking at apoptosis, or programmed cell death, of salivary acinar cells. So the feeling in the field is that irradiation just causes tissue necrosis. It just kills these cells. They're gone. There's nothing you can do about it. And that's the end of the story. Obviously, if you suffer from this problem, you have a diminished quality of life forever, because you can't speak, you can't swallow, diminishes the effectiveness of chemotherapeutic drugs. Your mouth becomes very susceptible to infections, and it's quite a health problem. Actually, there's also an immune system problem called Sjogren's syndrome, in which the immune system attacks the salivary gland, and you also lose production of saliva, and that affects 8 times more women than men. We don't know why. So we came at it from the perspective of saying, well, is it really tissue necrosis? Is something else going on? Can you block the death of these cells? Because we assumed it wasn't just necrosis. We assumed it was actually apoptosis that's being induced by the irradiation. And we come at it from the perspective, everybody has to have their favorite molecule. And mine happens to be an enzyme known as AKT, which is a protein kinase. It takes phosphate from ATP and puts it on proteins, on specific molecules. So AKT is protein kinase. It was actually discovered by a friend of mine who identified it as a gene that causes leukemia. And then he also showed that it was activated by a specific signaling pathway. What really got my interest was that if you take AKT and put it into cells and turn it on, it suppresses apoptosis. Now, normally we think about cancer in terms of growth stimulation, cells growing in an uncontrolled manner. But you can get the same point if you block the death of cells. So as a cancer biologist, that really interests me. And expression of activated mutants of AKT will block apoptosis that's induced by a wide variety of stimuli, including chemotherapeutic drugs. And AKT is very involved in signaling by the insulin receptor. It's a critical signaling molecule in the control of glucose levels in the body. So that was also interesting to us. It's regulated by phosphorylation of these two different sites here, and later I'll mention phosphorylated AKT. All that means is it's active as an enzyme. So if you take cells, tissue culture cells, that are salivary acinar cells, and you grow them in a tissue culture dish, or they're laying on the bottom of the plate, they have a very low rate of apoptosis. And what we're measuring here is how many cells are dead or dying of apoptosis. Even if you starve them with growth factors, they're quite happy for some time. If you stimulate them with a growth factor, and in this case we're using a molecule called IGF-1, or insulin-like growth factor 1, the cells don't die. In fact, they're protected from death. If you treat them with etoposide, which is a drug that's commonly used in chemotherapy, you see a dramatic increase in the number of dead cells. Now, if you give them IGF-1, this growth factor that can turn on AKT, it decreases the amount of programmed cell death, or apoptosis. And actually, if we take a mutant of AKT that's always on and put it in the cells, and now give them etoposide, it totally blocks cell death. Okay? Well, cells in tissue culture are, they're fun to work with, they're easy to work with, but they have nothing to do with real biology in most cases, okay? And in fact, they can't make spit. So if we're going to talk about a biological process, we've got to step it up. So we move to rodents. In fact, you can use mini pigs, rats, mice. Mice are easy because they're small, cheap, and we can do a lot of things genetically with them. So we've utilized transgenic mice. We've taken mice and expressed this constitutively active form of AKT, and we happen to have mice that make it specifically in the salivary gland. It was kind of an accident of our research, but we got them, we know to look for them, and so we found them. So we take these mice and we say, okay, if we irradiate them, what happens to them? So actually, when we do this, we wrap the body with lead, so we only irradiate the head and neck region, so it's just like a human. And then we look at what happens to either normal mice or transgenic mice. So here's the amount of apoptosis you find in a normal mouse, virtually undetectable, less than 0.5%. Take a normal mouse and irradiate it, and we're using 5 gray, which is about 2.5 times the dose you'd normally give to a patient in 1 day. Then look 8 hours later, we can find that about 8% of the cells are apoptotic. And if we look 24 hours, it's almost 30% of the cells are apoptotic. That is, they're undergoing programmed cell death. Okay? If we look at our transgenic mice, given the same dose, it's clearly much lower. It's about 2% here and about 8% here. Well, does apoptosis have anything to do with the production of saliva? Okay? Because like I said, the thought in the field was it's all about necrosis, and apoptosis doesn't really exist in the salivary gland. So actually here shows the detection of these apoptotic cells. This is so-called tunnel assay. So every brown cell is actually undergoing programmed cell death. So here's 24 hours after giving 1 gray, that's 1/5 the dose that I showed you before. Here's 48 hours, the dead cells are clear. But here's our transgenic mouse. Clearly there's a huge difference in the induction of these so-called dead or apoptotic cells. So what happens to production of saliva? So we irradiate the mice, and actually one group of mice are not treated with IGF-1, the others are injected with IGF-1 intravenously immediately before radiation. Then we look at production of saliva. So we inject them with a drug that makes spit flow easily, and we collect it in the mouth and measure how much is there. So if we look 3 days after irradiation, here are normal and transgenic mice that have not been irradiated. Here's a mouse receiving 1 gray of irradiation. It's down about 40%. 2 gray, which is the clinical dose, 60%. 5 gray, which is above it, down about 60%. In our transgenic mice, this response is clearly attenuated. So the presence of AKT, we believe, is protecting the salivary gland. Okay, so can we now do something other than make transgenic humans to protect them? Can we use IGF-1 because that is a potent activator of AKT. It's an FDA-approved drug for treatment of pediatric short stature. That is, children that are short and cannot respond to growth hormone. You give them IGF-1, and they gain height. So it's FDA approved. It can be given daily to children with no effects. It doesn't make them diabetic. It just helps them to become a little bit taller. Okay? So what if we do this to our mice? Well, if we do it with the mice, first of all, I can show you that we can actually detect the activation of AKT in the salivary gland. And in fact, we can detect it within 5 minutes after injection into the blood. So it gets to the salivary gland very, very quickly. All right, now what happens if we irradiate these mice? Okay, so we're looking at production of saliva 3 days after irradiation. Here's an untreated normal mouse. Here's a mouse given IGF-1 and not irradiated. Here's the mouse irradiated with 1 gray, which would be half the clinical dose. And you can see that there clearly is a profound effect on production of saliva. Now, if we just give them IGF-1 immediately before irradiation and then irradiate them, the mouse is totally protected. The salivary gland doesn't show any effect whatsoever. Okay. 30 days out, because remember, this is a lifelong change. Okay? So here's normal, here's an IGF-1 injected, the picture in black. 30 days, this is the normal mouse, just irradiated, almost 60% decrease in saliva production. Pre-treat with IGF-1, production of saliva is exactly the same. So what we're doing at this point is to, we've established that in fact irradiation does induce apoptosis, and the production of apoptosis is causally related to the salivary gland hypofunction. We've shown that if we express this activated mutant of AKT, it actually suppresses apoptosis and it protects the salivary gland from hypofunction. We can administer IGF-1 and achieve the same effect. And actually IGF-1 is better than the transgenic mouse. That's probably because it also stimulates some proliferation in the cells. So currently what we're working to do is establish the mode of administration. We're trying to use an oral administration, kind of like a mouthwash, as opposed to intravenous injection. Timing, can we give it how far before, or how can we give it after radiation? We're trying to determine a therapeutic index. In other words, IGF-1 can have an effect upon the tumor as well as the salivary gland. And so in doing these studies, we actually have tumor cells that glow green, they make light. And so we inject these tumor cells into the head and neck region, and we can quantitate how many tumor cells are there. We can irradiate the mouse. We can look at the effect on the salivary gland, and we can look at the effect on the tumor, both in terms of long-term tumor growth, but also in terms of acute effects upon signaling of apoptosis. And like everyone else has said, this involves a lot of different people. In my lab, Kaylee Schwartfigger actually generated the original transgenic mice, and Kirsten Lindison, who's a former postdoc, now has her own lab, who did a lot of the studies in terms of the effect of AKT and IGF-1 on salivary gland irradiation, and apoptosis, and hypofunction. Andrew Lewis is a technician in my lab who handles our colony. And we have an ongoing collaboration with Mary Raylan in the School of Dentistry and a postdoc in her lab named Greg Banniger. So with that, I'll be happy to answer your questions.